Alright, hello everybody. Today we're taking a look at lecture 11, which is talking about transcription. Part 2, actually. We're talking about RNA processing specifically today. So let's get into it. I already talked about lecture 10, go ahead and watch that if you want to learn about that. But let's go ahead and move into processing. So, basic gist of this, there are a couple different types of post-transcriptional modification or RNA processing, post-transcriptional, not to be confused with post-translational modifications when we get there. But we've got different types of RNA processing. Here are 3 different types. We've got a chemical modification with the 5' cap, which is basically just a guanosine. They're adding a guanosine cap to the RNA. It helps with stability. There's splicing / deletions, and then there's additions of poly-A tails and stuff like that. And then these types of processing, they're not limited to mRNA. They also happen in other types of RNA, but we usually associate things like the 5' cap and the poly-A tail with things like RNA. Okay, so 5' capping. You don't really need to know all of this, I think, but what I would try and kind of I guess, keep in mind or kind of memorize is we got the carboxy terminal domain of the RNA polymerase, right? Basically what's going on is here, I would try and memorize some of these. So TF2H, I believe, is one of the actual transcription factors you guys have, the general transcription factors, if I'm not mistaken. This, basically what's going to happen is you're going to phosphorylate the carboxyl terminal domain of the polymerase, right? And, this is while transcription is happening. So transcription starts, right? Transcription starts up here, and then you're going to phosphorylate these things, right? And, transcription is still happening, right? But, when that phosphorylation does occur, right, these negative elongation factors, these repressive proteins, are gonna bind the polymerase and arrest transcription, right? And when transcription is arrested, right, by the negative elongation factors, right, essentially what's going to happen is this capping enzyme, this green enzyme, is going to come in, and it's going to add a 5' cap to the end of the RNA strand that you're making, and then you're going to phosphorylate again, right? So I think here, hold on. Let me erase all this stuff. Basically, right, I feel like I made that way more convoluted than I needed to. From the start, right, you have all your transcription factors that are bound, right? This TF, so you're going to transcribe, right? And you're going to keep transcribing, right? And then TF2H is going to phosphorylate the C-terminal domain, like this end here. And then after the phosphorylation has occurred, I'm going to keep transcribing, right? Until I make some amount of RNA, right? And then when that RNA starts to poke out, right, the negative elongation factors are going to bind. And that's going to arrest transcription. Then the capping enzyme comes in, right, and caps the RNA, right? And after that, another enzyme, the PTEFB, phosphorylates the serine, again, serine 2 in this case, not serine 5, of the polymerase, and elongation continues, right? I don't think you have to memorize all of these. I would say, if you're going to memorize anything, try and memorize the phosphorylation, right? So phosphorylation here, right, allows for the binding of the negative elongation factors, and then the capping enzyme binds, which adds the cap. And then the PTEFB comes in later, phosphorylates again, and that causes the resting proteins to leave and elongation to continue. the one thing you should keep in mind is, here's a structure of what that cap looks like. It's actually just a guanine. It is literally a guanosine, a 5' guanosine cap. So Gu, I remember, likes to ask these questions where, which location has the 5' cap? And if he's asking for that, he specifically asks, which of them have, a modified guanine cap? Or, which end has a modified guanine or something like that. The answer is the one that has the 5' cap. So the 5' end of the mRNA should have it. And the other thing to keep in mind is this capping complex. It specifically binds to RNA polymerase. It only caps mRNA, and it has to wait for the 5' end to emerge. It cannot cap unless the 5' end has come out. And, the enzyme works basically by doing this, phosphohydrolysis reaction where it removes the 5' phosphate of the 5' phosphate of the DNA, of the RNA, not DNA, of the RNA, and then it does a nucleophilic attack where it takes this modified guanine and attacks, right? So it attacks the guanine, or this guanine phosphate group attacks the triphosphate, and then it kicks out the blue phosphate, right? So it's this O, right, had to attack a triphosphate up here initially before it became this kind of structure. And then there's also an O-methylation. I would memorize this. I'd memorize the N-methylation and the O-methylation. So the N-methylation, right? And then I would also memorize the O-methylation of the 2' hydroxyl, the first 2 nucleotides. So these should be methyls, right? You see how the arrows here? These two right here, those two O's are actually they should be methylated. So the 5' cap, this experiment is a very simple way of saying it's important for stability. So when we have the 5', the capped mRNA, what you'll see is, if you look at this, right, we have a lot of the mRNA is actually bound to the ribosome, and then there's a decent amount of that's unbound, right? So if it's bound to the ribosome, that means it's going to go through translation, right? Which is great. But when we remove the cap, all of it is unbound, right? That's all I'm trying to say. So basically, it's important that the 5' cap is present, because if the 5' cap is present, that allows us to actually do translation. If we don't have the 5' cap, we can't do translation. We also have the poly A tail, right? So the poly A tail is also helpful for stability and translation. This is, to add the poly A tail, we need a multi-protein complex that has a lot of different binding factors. So it has to cleave the 3' end and then add the poly A tail, essentially. So, give me 1 second, gang. Started dying in the middle of the freaking of the recording. All right. Anyways, yeah. So now back to this. So what you should know from this, right? Is that basically what happens is we cleave the 3' end of an RNA to add the RNA, the poly A tail. And the enzyme that does this is the important part, is PAP. Polyadenylate polymerase, which is a template-independent RNA polymerase. It does not need a template strand. It's also an RNA polymerase, not a DNA polymerase. And, what this does is it basically helps protect the mRNA from being degraded and stuff like that. So here's an example of, what's going on here, right? For this we have so here we go. So if you look at this, right, see this massive complex here. So polymerase 2 transcribes the poly A addition site. So basically what's happening here is that polymerase 2, right, the RNA polymerase, right, is going to transcribe some stuff, right? It's going to transcribe the RNA, and then it's going to make this thing, right? And then what's going to end up happening is it's going to run into this like AAA sequence, and this multi-protein complex is kind of able to bind to that, right? And then when that happens, the polyadenylation factor binds this sequence, and that initiates the mRNA cleavage. So essentially what's going on, right, is first the RNA polymerase has to transcribe the sequence that the polyadenylate factors and the PAP actually recognize, right? Once those are transcribed, then the PAP and all those factors are actually able to bind to that. And when they bind to that, what that results in is that the PAP binding and all those factors binding causes the mRNA cleavage. So it actually cleaves the 3' end of the strand through an endonuclease activity. And then after that's cleaved, PAP adds the poly A tail, and then we also add some polyadenylate binding proteins, some PABPs. And those PABPs are basically there to stop the poly A tail from degrading itself. So they help with stability. They help the poly A tail from stopping itself from degrading. So that's kind of the purpose of that. We also have, intron and exons and stuff. So briefly on an exon and an intron, I'll talk about that. So an exon is a sequence of a gene that can be transcribed and turned into an mRNA and then can be turned into a functional protein. An intron does nothing. It's just a region of RNA that doesn't do anything, or a region of DNA or whatever, right? So, but it ends up being removed before translation happens, because actually if you don't remove them, it actually ends up with a messed up protein, basically. And splicing is the process of removing the introns and connecting exons. So you can imagine, right, so if exons code for, like, proteins, right, and introns code for nothing, you could, like, mix and match different combinations of exons. So, like, in this case, right, we have exon A, exon B, exon C, and stuff like that, and then we have the introns, right? We can splice out the introns. Basically, I could have a case where I have a mature mRNA that has A, B, and C. Maybe I just have B and C. Or maybe I just have C. Or maybe I have AB and no C. Whole bunch of combinations. Maybe I have AC. There's a whole bunch of stuff you can do with this, right? So there's two types of introns as well, by the way. There's self-splicing introns and spliceosomal introns. So self-splicing, they splice themselves out. And spliceosomal, they need spliceosomes. I'm not going to go over this really. I don't think it's that important. But yeah, if you want to take a look at this more, you can take a look at it in your lecture. But we're going to talk about splicing. So this is specifically for self-splicing introns, which I think they refer to as group 2 introns, which is weird because they don't really talk about group 1 introns at all. But these are self-splicing introns, okay? Basically what's going to happen is we have our pre-mRNA, right? This is the mRNA before any splicing has happened, remember. Remember, the splicing occurs at the mRNA stage. It doesn't occur at the DNA stage, it occurs at the mRNA stage. It's part of mRNA processing. Basically what's going to happen is, for the self-splicing introns, there's going to be some part of the intron, right, that has an OH group, right? One of these 2' OH groups. And this 2' OH group, what it's going to do is it's going to attack itself, basically. It's going to attack, it's basically going to, like, what's it called? It's going to condense, right, and form this bubble first, right? And then when it forms this bubble, right, this bubble basically allows one of the 2' OHs from the intron to get close enough to the other side of the intron to the point where this OH is able to attack one of the phosphates, right, in the backbone of this mRNA. So you can imagine what's happening, right, is like this is going to attack, right, like this arrow shows here, this arrow shows, right, and then it's going to basically kick off the electrons to that. So you end up with this kind of like if you will, like this, right? And then this is all going to connect as one, right? Something like that, right? Kind of drawn poorly, but you're going to end up with what's shown in this step, right? And now you just have two separate strands of mRNA. But now what's going to happen is that the now remaining 3' OH from the section you just removed, right? Because remember, this U, right, has an OH group now because we just removed this phosphate. It's going to attack this, and it's going to reform the completed strand, and this is just going to leave as a leaving group. And then here's an example of what's actually happening. So here we have like the intron, exon, and 3' exon. We've got the 5' exon, and then we've got the intron, right? So these two yellow things are part of the intron. So essentially what's happening, right, is when this OH attacks, right, this phosphate leaves. And so when that all happens, right, you end up with this, this is going to become an OH, or like an O-, right? And this yellow portion here is going to form like this circular structure, right? Because this OH is attacking here. It's going to form this like circular structure, if you will. And it's going to break off. It's going to like break off from here. And then what you're going to have is you're going to have a nucleophilic OH, that's this. And then you're going to have the 5' phosphate that's not shown, but there's like a phosphate here, right? And so what you can end up doing is like once this OH is there, you can use this OH to attack the phosphate that's right here, and you can reform your strand, basically. It's a transesterification reaction, right? Where you have a phosphoester. And, you know, like I said before, it's important to align the nucleophiles. Like, you know, with this alignment thing, you know, it's important that, like, this kind of happens here. Like, this condensing happens to align the nucleophile. So this nucleophile is close enough to that U to actually do the attack. And, you know, cell splicing, they can fold into each other. This should make sense, right? It's like what happens here, right? This is, like, folding on itself, if you will, right? If you look at this, like, see how it's, like, condensing like that? It's folding on itself, right, to make it easier to splice. So, you know, if the splice sites are here, right, you can imagine, like, this is a massive RNA, but, like, maybe if the RNA has a lot of intermolecular base pairing, or intramolecular base pairing, actually, a lot of intramolecular base pairing, right, it's going to, like, base pair to itself a bunch and cause these two sites to become very close to each other so that this kind of OH mechanism can happen, right? So, like, maybe there's, like, a OH, there's, like, an O here, and, like, the phosphate, like, attacks the phosphate. This attacks the phosphate here and stuff like that, if you know what I mean. So the other type is spliceosome requiring splicing. So these require a protein called a spliceosome. And the spliceosome is a ribonucleoprotein that basically means that it contains RNA and protein. And I hope this makes sense because you can basically what these guys are doing is these snRNAs, these small nuclear RNAs, right? What they do is that they have these like they have base pairs, right? Because small nuclear RNAs, they have base pairs, and they are able to bind to the intron, right? So you see this, like, it's able to bind to the intron, right? See this, the yellow part here. And there's many of them. So this is an example of a U1 snRNP. It's looking at this guy. But there's 1, 2, 3, 4, 5. There's 5 in this picture alone, right? But there's hundreds of them, thousands of them, right? And they all have these little sections of snRNA that are able to bind to parts of the intron. And so what happens is when they bind, right, they use these SNRNPs, these ribonuclear proteins. They use the intron to bind to the they use the intron to bind to the they use the snRNA. They use the snRNA, right? The complementary sequence of the snRNAs that are in the introns to bind to the introns, right? Because these snRNAs are complementary to the introns. And because we have so many of them, these all bind to the intron. And then what that does is these all act together and they basically cleave. The proteins, right? So notice it's a ribonucleoprotein, right? There are proteins as well, right? All these like other colors, these green things, purple things. Those proteins act to cleave the bonds between the intron and the exon. So they allow for cleavage, basically. So basically, what you should get from this is that the snRNAs, they're used to identify the section that needs to be cleaved using H bonding, right, and complementary base pairing, and the proteins themselves do the actual cleavage. That's the main gist of that. I'm not going to bother going through this. I'm just going to explain this, right? The purpose of alternative splicing is that you can make many different versions of a protein, right? So if I have exons A1, A3, B1, B3, C1, C3, I can combine these in so many different ways, right? So you can see here, like, I have A1, maybe A1, A1. But you can see here, like, I've changed the C exon, right? This is 1, this is 2, this is 3. So basically, alternative splicing allows us to combine many different exons together, right, in different, like, formats, if you will, to get different protein structures, right? So it's basically just a way of, making variation, right, in our proteins. And in the math here, right, it should hopefully make sense, right? So in this example, right, there are 3 different options for my A's, right? So there's 3 of them. There's also another 3 options for my B's, so it could be any of those 3. So it's going to be multiplied by 3. And there's another 3 options for my C. It could be any of those 3. So the whole total amount of combinations I have is there's 27 different combinations that I have, right? So there's 27 different combinations that I could have for just having 3 genes, right? These are only technically 3 genes, right? So it's basically just a way of making variation, right? So I can take, A1, this guy here, attach it to this, and then attach it to this. Or I could take A2, attach it to B1, then attach it to C3. I don't know. You can make it up, right? You can do all sorts of things. So a whole bunch of different combinations, essentially, is what I'm trying to get at. And this is just an example of... This is not very important. He's just showing an example of alternative splicing. Not that important, though. And this is actually a pretty cool slide. It just kind of talks about how alternative splicing is actually very important for neurons to stop synapse. Basically, it's very helpful for neurons because it's the mechanism that allows them to not synapse on themselves. But basically, like, for neuron A, right, like in this case, in order for... Let's see here. Basically what happens, right, is for neuron A, right? Neuron A is going to make some spliced versions of these DSCAM proteins, right? So DSCAM is some sort of it's expressed on neuron surfaces for homophilic interactions, right? And then dimerization, so it's on the membrane of the neuron, right? And then dimerization of the same DSCAM triggers self-avoidance due to neuronal growth. So basically, when these things dimerize, right, it stops the neuron from synapsing on itself, right, or growing on itself. So what's happening here, right, is neuron A, right, is going to produce some spliced versions of these proteins. So IG2, IG3, IG7, right? All neuron A's produce the same proteins there. So if 2 neuron A's come in contact with each other, or one neuron A comes in contact with itself, right? Neuron A is always going to have the IG2, IG3, IG7, right? So it's going to have that. When these bind together, like this says, dimerization, right, triggers self-avoidance, right? So these are going to bind, and it's going to cause self-avoidance, right? Because they're dimerizing, right? It's going to cause avoidance. So A will not bind to A. But A on B, right? Remember, A has these, but B has something completely different because it had different splicing, right? And so because these cannot bind to each other, right? They're given the green light, and they're able to synapse onto each other. This is just a slide that's saying, antibodies use the same thing. And, there's a bunch of different antibodies in our body. These are not that important, I don't think. So I'm actually not going to go over these. If you really want to look at these, feel free to watch the lecture again. But these I don't think are very important for your exam, so I wouldn't worry about them. But yeah, I hope this video was helpful. But yeah, the next content is going to be quite challenging. Translation is very dense, but yeah, again, I hope these videos are proving to be helpful. If you have any questions, drop them in the comments, or come to the IC and ask me in person. But other than that, good luck studying, guys, and yeah, have a good one.